Hi, everybody, and welcome to Chapter 1 of Microbiology. Let's just go ahead and get right into it so you guys aren't having to sit here for too long. All right. All of the chapters are always going to start with our learning outcomes. That basically just means that that's what we're trying to target for what you guys are learning for this chapter. Some of them are going to have more, some of them are going to have less. So if you guys are interested in what you should be learning for each chapter, that will be listed in the beginning of the slides for each one of these. I'm not ever really going to read them to you because I figure like they are there for, if you're interested about what you're supposed to be learning, but hopefully you're learning it while I'm teaching it. So if you need a reminder of what you're supposed to learn from it, you can always refer back to that. Okay. Microbiology. So you guys are here to learn about microbiology, and it has a lot to do actually with healthcare, not just human healthcare, of course, veterinary healthcare as well. So many diseases are communicable and are caused by microbes, right? So microbiology is the study of those microbes. Basically, a microbe is any organism that can't be seen with the naked eye. If we have to use magnification in order to see it, that's a microorganism. So we just abbreviate that or shorten it down to microbes. We've also referred to them as germs or bugs. That's fine as long as you know what you're talking about there. But that's microbiology in a nutshell. The major groups of microbes that exist are going to be the cellular microbes and the non-cellular microbes. Yes, and by non-cellular, I literally mean they do not have cells at all. So the cellular microbes, that's going to include the bacteria, the archaea, the fungi, the protozoa, and the helminths. And then we have our non-cellular organisms. That's going to include the viruses. Yes, they do not have any cells. That's the truth about viruses, no cells at all. And then prions, those are just a protein that causes an infection. We'll get into a little bit more detail on those in a later chapter. Microbes, okay, they grow up really quickly. So you guys are going to be doing this in the laboratory as well. We're going to be taking sterile media. It's a broth. It's not unlike soup broth, but we're going to be inoculating that sterile media. And that just means introducing new microbes to that sterile media. And then they're going to grow up really quickly. We would have enough of them, like say the next day. So it's pretty interesting. Well, they'd be better, right? Because that's whenever, summer people only have till the next day. They don't have 2 days to work on this stuff. So, but yes, they grow very, very rapidly. We can make changes in them and see the effects of those changes very quickly, which makes them very useful for genetic studies. However, we cannot analyze them directly because we cannot see each individual cell directly because we need to use a microscope. And so you might say, well, we can't see, people in their individual cells either. No, you're right. Okay. We can't see people's cells individually. But if I take a cell from a person's body, one cell from a person's body, or multiple cells from a person's body, and then separate it from that body, it doesn't live separated from that organism. So that's the difference between a multicellular and a unicellular organism. We have to have all the cells together in order for them to be able to survive. Case in point, if I lop off your leg, that's not going to live very long, detached from your body. So these organisms, these microorganisms here, many of them being unicellular, one cell, we want to be able to see them as an organism. So a single organism would be one cell for these guys. So microbiologists have a lot of different things they want to study about microbes and would be interested in understanding maybe their cell structure and function, the growth, the physiology, and the genetics of these organisms. And these are actually pretty important as far as I did not select hold on. These are pretty important as far as the medical field, sorry. So cell structure and function, growth, physiology, and genetics. This applies to what we do in medicine now. If we can manipulate these things or target these things with drugs even, that's important to know for medicine, right? We want to try to figure that stuff out other than just trying to study it for, knowledge's sake. Taxonomy and evolutionary history. This is not something I am particularly interested in, but it is something that microbiologists study that can help us understand where we came from. taxonomy is basically organizing things based on similarities that could be genetic or they could just be observable similarities. And then we have interaction with living and non-living environment and how microbes can affect whatever environment they could be living in, whether that is going to be causing disease within their host or changing the environment that we live in, which we'll get into more detail with in a moment. That's what the whole point of this chapter is. So there's several different branches of microbiology that result from studying these different aspects of how microbes interact with us. So medical microbiology and epidemiology are pretty hand in hand, and immunology, of course, sneaks in there as well. And then definitely going to have the most to do with what you guys are trying to get into in healthcare, compared to the others, industrial, agricultural, and environmental. So environmental, of course, microbes and their role in the environment. Agricultural, especially important as far as the soil and the health of the soil, especially nowadays with climate change. And then industrial microbiology, just large scale production of, whether it's food and trying to make sure that the food is safe for you to consume, or the actual production of things made by the microbes themselves, right? That could be anything. Whether it's insulin that we've had them engineered to make, or that we're using them to make bread, right? So the top three are the ones that you guys really would be the most interested in: medical microbiology, looking at disease-causing microorganisms, public health microbiology and epidemiology. That became really important with the introduction of COVID in our society and trying to determine where it came from and how to keep these things under control. That's a lot of what epidemiologists will do, trying to keep infection spread under control. And then we have immunology. And for those of you that don't know, immunology has to deal with studying of the immune system. And we are going to get into the depth of how the immune system functions in a later chapter. I think it is one of the most fascinating things about the human body, if not the most fascinating thing personally, my opinion. But we will get into, lymphocytes and leukocytes and the difference between those and their actual functions and what they do for the body in defense of microorganisms as well. Microorganisms, they've shaped the earth. I don't know really how to describe it other than we have eukaryotes, which are organisms that have a true nucleus, right? So a nucleus, that just means that's where our genetic material is made, or sorry, not, well, it is made there too. So I guess that's not wrong, but that's where it's stored in the cell. And there's a membrane around it in a eukaryote. It's a true nucleus. That's what that word means. Eu meaning true, karyo means the nucleus, okay? And eukaryotes are the ones that don't have a true nucleus. So in bacteria and archaea, their genome just kind of floats around, and it's in a region called the nucleoid region, that where it hangs out, but to keep it away from everything else, to not get caught up in everything. That's nice. But they don't have a true nucleus. It's not membrane bound and contained, and they don't have organelles like we do. So what's an organelle? That's like your mitochondria. So that would be your powerhouse of your cell that makes your energy. They're like little organs. The organelles are little organs in your cell that have specific functions for the cell. Eukaryotes don't have those. They are literally the only membrane they have is going to be the one surrounding their outside. There's no like division inside. Okay. Right. So it would follow and make sense then if I were to tell you that evolutionarily speaking, whether we look at the actual DNA evidence of this or just looking at the cells and how they function, that the eukaryotes, A being lacking a nucleus, right? Eukaryotes. Those guys came first evolutionarily. And then eventually we had the evolution of eukaryotes, like more than a billion years later, FYI. So these eukaryotes have been around a lot longer than we have. Okay, so we are eukaryotes, right? And there are other microorganisms that are also eukaryotes. Not every microorganism is an eukaryote. The bacteria and the archaea, which look similar under a microscope, but actually are very, very different from one another. Those are the eukaryotes, okay? Eukaryotes, like bacteria and archaea, their cells are much, much smaller than the eukaryotic cells, typically. So what is evolution? We talk about evolution and how eukaryotes evolve from the eukaryotes. Evolution has to deal with the accumulation of hereditary changes that occur in an organism to adapt to their environment. So just because you are the new, you had this new idea to try this new thing and, it worked out to have a better life for you. If it's not hereditary, who cares? You're not spreading that to your offspring. It's not going to benefit your whole species or something. Evolution says that in order for it to be considered, a new change and count as evolution, it has to be hereditary. So these adaptations that have come to be are usually a result of random mutations. And you might be sitting there like, random mutation is going to lead to like these beneficial things. That doesn't make any sense. That would take too long. Not in a bacteria. I can show you in probably less than 24 hours, bacteria that will randomly mutate and become spontaneously resistant to antibiotics in less than 24 hours. So you would be wrong. So there is evolution, it's been documented in all corners of the planet. Like is saying here, and we've seen it, it's testable with science. We see it in the microbes. They give us the proof that it is something that's happening. It's way more difficult to observe this in larger animals that take a lot longer to reproduce, but it is there and it does happen. We're talking about over not a period of 10 or 100 years. We're talking about, millions of years, billions for, eukaryotes. Bacteria, so they started out being the first ones that could go through photosynthesis. And if you guys know about plants, you know about photosynthesis probably. You might not know every single intricate detail about it, and that's fine. But plants, photosynthesis, basically that's how they take the carbon dioxide from our atmosphere and turn it into oxygen for us. And that's like a real loose breakdown of what's going on. But yeah, so it's one way that they are able to drive making energy and building new molecules for their cells. So basically, bacteria did it first, and they used to do it in a way that they did not make oxygen, but eventually changes in their genome and evolutionary changes enabled them to then make oxygen as a product, and then other organisms could come into existence as oxygen was built up in the atmosphere and could be used more effectively by larger organisms that needed oxygen to survive based on that change in the environment. So that literally helped change the planet. We did not have an oxygen-rich environment before microbes. Bacteria did that for us. The whole reason that we have all of this going on with the plants and the animals and everything, that exists thanks to microbes, because this atmosphere didn't exist before them. I think that's pretty cool, I guess. It's not that exciting, but it's, I guess, nice to know. So bacteria, even nowadays, bacteria and other microbes like algae are the ones that more than 70% of our photosynthesis on the planet is done by those guys, not plants. Bacteria and algae. Algae are not plants. Algae are not plants, okay? Of course, bacteria and fungi, which are other microorganisms, those are the ones that are going to be involved in breaking down dead matter, waste, things like this. All of that is done by microorganisms, and we need them to do that. Otherwise, these things would just build up on the planet and not go away. Of course, if microorganisms didn't exist and something died, it wouldn't smell bad because there would be no rotting. So I don't know. Well, you want to look at it that way. I don't know. I don't know. We probably shouldn't be talking about that. It gets real dark real quick. So, right. We just talked about the gas production of microbes as far as in photosynthesis and how they were the first ones to start making oxygen in our atmosphere to change it. But we also have organisms that are creating methane in methane pockets, like maybe in swamps or something like that, because these environments are now, they lack oxygen more. So now we're producing more methane as a product of having to live without oxygen, not us, but the bacteria that live in these areas due to climate change and all of this. And it's just making it worse because they're just adding more and more to it and everything like that. But they have a lot of other effects on our environment as well, as not all bad. Okay, so we obviously have used microbes for years and years and years to help with make our foods, or leading into making antibiotics. Our first antibiotic, penicillin, that basically was made from observing mold. And we noticed that not we, but it was observed that the mold, bacteria wouldn't grow near the mold. And so it was found out that what was being made by that mold was called penicillin. And they used that to treat bacterial infections, which is neat because it's naturally made by the fungi to keep the bacteria away from whatever it's trying to eat or wherever it's trying to live and everything like that. So that's just its defense mechanism on its own. And we were able to use that. Apparently in Egypt, they figured that out too. They just didn't know how to isolate it and use it that way. So we just use moldy bread itself to treat, wounds that seem to be becoming infected. So interesting little fact there. And of course, we know that microbes drive the production of our beer, wine, bread, and cheese. We'll talk a little bit about that later. What kinds of other uses? Nowadays in modern society, we have biotechnology for, whether it is using microbes for helping us in medicine or reproducing like proteins that we can't make for ourselves. We can engineer bacteria to make these proteins like insulin and utilize those proteins and purify them either for studying or other things like that. And we can use them to study all sorts of genetic abnormalities that we wouldn't be able necessarily to study in people, to understand our own biology a little bit better, which is pretty interesting since these guys grow so quickly. They're a nice tool in that way. Then we have, of course, bioremediation. So we talk about recombinant DNA technology. Just take a step back to that one really quick. Recombinant DNA technology, we're literally going to be taking like genes from one organism and putting them into another organism. So it could be from like one bacteria into another bacteria, but or it could be from different animals, or it could be whatever, cross species, whatever, and trying to alter these organisms for whatever benefit that we might be looking for. That's recombinant DNA technology. And yes, that is where we're getting the idea of GMOs, genetically modified organisms or transgenic organisms. We will investigate that a little bit further later on in the class. This is all just real basic concept introduction. And bioremediation, coming back to that, is going to be getting rid of toxic substances that otherwise couldn't be broken down or would take a very long time to be broken down. We can engineer bacteria to come and break them down faster. Things like oil spills. We know microbes can cause disease. We know that. We've seen COVID. We know about pneumonia. We know about a lot of different things that can be caused by microbes. These microbes that cause disease-causing microbes are called pathogens. And a disease is any deviation from health. So if you have what we consider normal health, so if you have any deviation negatively from that state, that is a disease. So of course, not all diseases are contagious. Those that are, we consider probably caused by microbes. Like cancer is not a communicable disease for the most part. Certain cancers can be, but just in general, speaking about that, heart disease and stuff like that, not microbes there, right? So these are the most common causes of the top causes of death in the United States. And next, we're going to show a graph for the entire world, just so you can keep that in your minds of what's coming up, so you can compare the two if you want to. But we have heart disease, and then cancer, and then COVID in the United States, that you're more likely to die from COVID than you are, from an accident, having an accident like a car accident or something like that. But yeah, even stroke is lower, diabetes is lower than COVID still. So that's pretty interesting. That's the United States though. In the whole world, it doesn't fall quite so high up on the list, COVID. We have heart disease, stroke, chronic lung disease, then influenza and pneumonia before COVID, but they're pretty close to one another, right? You can see that on the graph there. I want to be clear about pneumonia. Pneumonia is a pretty broad term, right? Anytime that you have, an infection in your lower respiratory tract that is having, fluid collection in your alveoli, the little sacs in your lungs, then you have the development of pneumonia. What kinds of organisms cause pneumonia? What bacterial or viral or whatever, what specific organism causes pneumonia? And I want you to rack your brains too much about this, because you're probably going to be wrong. Because the idea is a lot of different things can cause pneumonia. That makes pneumonia something called a syndrome. So a syndrome is where you see pneumonia. These are the symptoms for pneumonia. And we know what pneumonia is, but it can be caused by all these different things. We see that with meningitis as well. So it's not only caused by the meningococcus. It's not only meningococcal. We know that meningitis can also be caused by other bacteria and as well as fungi. We can have fungal meningitis. So moving on. I just want to bring that up since it's not any one specific pathogen, right? Remember, pathogen is the thing that causes a disease. So bringing up one particular pathogen that's particularly difficult for the world to manage, especially in third world countries or undeveloped countries, we have malaria, right? Kills about 450,000 people every single year. It's transmitted by mosquitoes. A mosquito bites a person. The mosquitoes themselves are actually infected. They're not only going to be transmitting the disease from one place to another, they are actually infected with the malaria protozoa. That malaria is caused by a protozoa, by the way. And we will talk more about it. Don't worry, don't worry. The best way that we can prevent this is through the use of nets, which are not very expensive, but even these poorer families, what we call these smaller tribes and groups in sub-Saharan Africa, they still can't even afford that. There is a vaccine available. It does not protect very well, and people aren't going to keep up with the boosters in these areas either. So it's just hard to control stuff like this. And that puts things into perspective as far as, an epidemiologist and how they would look at something like this and how do we control infections caused by organisms like malaria that are affecting, underdeveloped countries. How do you deal with something like that? How are you going to get rid of these diseases? Well, they can't treat them the same way that we do. We just have new versus old diseases, right? New. SARS is new. HIV is new. And when we look at things like polio and leprosy, obviously those have been around for a very, very long time, and they've mostly, not entirely eradicated. There's still some, of these guys hanging about in the world, but they're almost mostly eradicated, and they've been very well controlled. So, the longer the thing's around, the more likely it is that we're going to tackle it effectively. Of course, there are now discoveries that some diseases that we thought were not microbially related are in fact microbes that are causing them, like ulcers. Ulcers are caused by the bacterium Helicobacter pylori. Isn't that interesting? About half of the population has those. There's also been a link between Coxsackie virus and diabetes and schizophrenia. That's terrifying and very interesting. If you guys don't know, Coxsackie virus is the one that causes the hand, foot, and mouth disease. Think about that now. If you have kids, you're probably having a heart attack right now. Don't necessarily worry about it too much until you have to worry about it. There's nothing you can do about it if you can't do anything about it, right? So if you are concerned about it, go talk to your doctor. I'm not the person to bring this up with. If you think your kid's going to wind up a schizophrenic because they had this disease, I think the likelihood is very low because it's a pretty common disease, but there does seem to be a link with this. And then we look at multiple sclerosis, OCD, obesity, all of these other things linked to chronic microbial infections, things that are so low lying, you might not even know you have these infections. Interesting stuff as well. Now we're looking a lot more into stuff that is kind of similar to that, or the effects of having had an infection, for a long time, and might be quieter effects of it that we might not, have looked into. previously, but now we have the means to look into it and link it back to, the causative agent for these things, like cervical cancer being linked to human papillomavirus. We know HPV can cause cervical cancer. It seems obvious to us now, but how would we have discovered that if we didn't have the technology that we have now to look at something like that? Chlamydia can lead to female infertility. The imbalance of the gut microbiome can have a lot of negative effects on your body as well. So we have an increasing number of patients with weakened immune systems. Why would we have people with increased immune systems if the world is becoming more developed? Because, I'm not trying to put it in a bad way, but more people are surviving when in the past, like long ago, they wouldn't have. So if you had a bad immune system in the 1600s, you might have died from the plague. But nowadays we found ways to help keep you alive. So we have more and more people that have, weakened immune systems for that reason, or we have increase of HIV infection that might not be managed well. So then they have weakened immune systems as well. So they are more likely to have these diseases that are less common or opportunistic infections, which are infections that, wait for your immune system to be low and then they become infectious or pathogenic. Otherwise they wouldn't be. And that can lead to a rise in disease that we wouldn't otherwise see, as well as increased drug resistance, which you don't need me to tell you about if you know anything about MRSA. Microorganisms. The single-celled in general, right? In general, single-celled organisms are going to be bacteria and archaea, for sure. Those were two, always. There are some eukaryotes like protozoa that are also single-cellular organisms. Then we have some organisms that are multicellular, like we are. The helminths, that's like the worms, the infectious worms, and then the fungi. That's fungus. I don't know what to tell you. If you've seen a mushroom, then you know that they can be multicellular. Really basically, as far as their size goes and the organization of the cells goes, bacteria and archaea are much smaller in size, generally speaking, than a eukaryotic cell. And they would look like little tiny things on the outside of the cell. So here we have approximately, 10 times smaller than the average eukaryotic cell. So they're much smaller. They do not have any organelles. So those are those functional units inside of the cell, like the energy-making mitochondria and the photosynthesis performing chloroplasts. Those are examples. Organelles also are membrane bound. So they're, bound, the outside's covered in a membrane to keep everything together. Eukaryotes do have this, okay? So bacteria and archaea being eukaryotes, they don't have the membrane-bound organelles. Eukaryotes do have the membrane-bound organelles. They have the larger cells as well. Then we have the acellular organisms like viruses. Again, viruses are not going to have any cells in them at all. And then prions, which are infectious protein particles. Get into those in a little bit. Not right today, but another time. These are just some pictures of some of these organisms that we are talking about. We have the helminth, like the worm. This is like the head of a tapeworm. This is Tinea solium, which is the typical port tapeworm. Then we have fungus here. So we have here, this is a reproductive structure of the fungus. Then we have a protozoa, a bacterium, a virus. This is herpes simplex virus. And then we have a prion, which is just proteins. Okay, so back in the day, we thought disease came from spontaneous generation, which is exactly what it sounds like. They thought that disease is spontaneously generated, like you just became sick. There was no reason for it to be caused by it, or you smelled a bad smell and the smell was what caused it. So there might have been some other sort of molecular form of matter that would cause a disease. It was not at all considered that it could be caused by, another living organism that was so tiny that we couldn't see it. Pasteur did these experiments with sterilized broth in these flasks that showed that, if you sterilize the broth first, and then break the little, neck, I guess that's the word for it. But if you break the neck on that flask, then the bacteria can get into it. And they couldn't really get into the bent version as well. So this would remain sterile. But if you break it, it would grow. And so, that's how he proved that no, spontaneous generation is not a thing. There is actual living cells in here. Something is alive and can be sterilized and getting rid of. So, because we can keep something, if you had something that you sterilized, it makes sense to us because you're looking at that like, well, duh, it was sterile and then you kept it sterile, so it stays sterile. If it's spontaneous generation, that wouldn't help you. They would just spontaneously generate because spontaneous generation. So, anyways, microscopes were developed and then we were able to see these microbes. So Robert Hooke made first observations of microbes, and then Anthony van Leeuwenhoek was the one who developed the first crude microscope. Scientific method is used by scientists to explain the phenomenon in the world, right? Most of you guys have heard about the scientific method, so you know what a hypothesis would be, which is, an educated guess as to what might be going on. You make a guess to explain something that you've seen in the environment, and then you do tests to try to, prove or disprove. The results would prove or disprove that hypothesis. So you have you formulate a question, that's going to be your hypothesis basically. So you have like, what is going to be going on? What's going on with this disease? What is it caused by, right? So you kind of look into it a little bit further, you do some research. This is more or less like the modern way of looking at this. And then you have a formal hypothesis, like, okay, I'm betting that this disease is caused by this virus. And so now we can perform some experiments. You would call that performing diagnostic tests, but that's the same thing. That's the exact same principle of what the scientific method is. The scientific method is going to drive a lot of medicine. You probably didn't even think about that you were doing that. If you look at a patient and you think that they probably have the flu, and then you go and swab their throats, and then you do the PCR test, or you do the, rapid influenza test, or whatever you want to do, whatever kind of test that you end up doing. Guess what? You just formulated a hypothesis and tested it, and then you proved it. That's what the scientific method is. It's that simple. So don't overthink that. If, of course, you were wrong, if it came up negative, then you might want to try testing, I wonder what it is. I wonder if it's, if it's not the flu, is it COVID? And then you might test for that. Of course, you probably do both at the same time, but you get what I'm saying. So we have deductive reasoning and inductive reasoning. What I just explained to you is deductive. We are using a general principle to explain specific observations. In inductive reasoning, we are basically making observations just through general experimentation, and then we have the creation of a general principle based on that, sort of like a reverse way of looking at things. So inductive reasoning tends to be more like a Sherlock Holmes approach. So most of the time with the scientific method, it is going to be a lengthy process of experimentation, analysis, and testing. So we will either support or refute the hypothesis, but we don't just treat that, when we're looking at diagnosis, and these are, experiments that have been tried and true for a while, yes, that's fine. We go forward with your results, and that's that. However, typical scientific method leading to a hypothesis is not going to automatically create a theory or a law. Those things have to be proven over and over and over again, maybe by other people in like slightly different scenarios, that just to show repeatedly that this is how this thing is and how it works. So we don't automatically accept it as fact. So we want to have several people looking into it. Right. Yeah. So I don't know. This really just says, like, I feel like I'm more or less like what a theory is. So a hypothesis that you have, had your proposal, your hypothesis, you do experiments, and you proved it, you supported it. and so many other people, and there's this whole body of evidence, not just your single experiment or your one paper that you published. There's a whole body supporting this concept. That is where a theory comes from. And a theory is not a law. A theory is a theory. It's just a theory, and it stands the test of time, but is not a law, because we're admitting that there may be stuff about this that we still don't understand. So that's important to remember. Theories are theories. They're not rules. They're not laws. They're not, infallible. So, so a lot of these theories might change as we have gotten new technology available to test them, right? That's just kind of what this is more or less telling us. As technology has advanced, our understanding of certain theories has allowed us to change those theories. Think about it back in the day, right? How they used to think spontaneous generation, and that made sense to them. And then Pasteur came along and upturned all of that. So John Tyndall is the guy that came up with an idea of how to basically remove spores and viruses and things from broths or whatever you needed to remove bacteria from to make it sterile, like we do with an autoclave in a hospital or something like that. We want to make something sterile and remove microbes. John Tyndall came up with the concept of doing that and approaching things that way. Robert Koch is the guy that came up with an idea of different steps on how to prove what the causative organism was of a disease, the pathogen that caused it. Then we have Oliver Wendell Holmes. He noticed that mothers who gave birth at home experienced less infections than mothers who gave birth in the hospital. So why could that be? Well, Semmelweis will tell you. Turns out that women were becoming infected in the maternity wards after physicians who had performed autopsy would come and deliver the babies and not wash their hands. So they came up with the concept, maybe this is what's causing this, people getting sick. So they linked it there. Then we have Lister to thank for aseptic technique, utilizing hand washing and misting in operating rooms in order to remove as many of the microbes as we possibly could. And now that concept has stuck with us in medicine and pretty much in anywhere that you're performing microbiology, even to this day, right? We know washing our hands and all that, how important that can be. Pasteur also invented pasteurization, so heating and cooling liquids so we don't damage the liquid, but can get rid of microbes or at least pathogens in the process. He also was the one that showed us that human diseases could rise from infection. So Pasteur, we talked about him just earlier. That was the guy that had the necks on the glass with the boiling solution, and it was supposed to be sterile, and then they broke it, and then the thing grew, and no such thing as spontaneous generation. That's Pasteur, right? So Robert Koch, we've just talked about him as well. He was the one that showed the steps on what to prove what pathogen causes what disease. Together, these two, essentially, their work supports that idea of the germ theory of disease, that diseases are caused by something communicable, right? If it's communicable disease, there has to be something communicable there. So that is the microbe. So that's the germ theory of disease. Germs cause disease. Go figure. All righty. Let's get into naming and other boring things. So nomenclature, that's just how we name an organism. We have a rule called the binomial nomenclature on how we name organisms. That's just like Homo sapiens, Staphylococcus aureus, those things, which we'll talk about in just a moment. There's literally the next slide, okay? Classification, arranging organisms based on observing them or their actual genetic makeup. And then identification, we can use that taxonomic scheme to break down essentially what organism you might be working with based on, the further down you can get into and deeper, more specific, you can get into the taxonomy of it based on their traits and their genome and everything like that. It's useful for identification, I guess. So that's kind of what taxonomy is there for. Now, binomial nomenclature being naming, okay? So the binomial system of naming. It's literally just going to always be capital first word. Capital first word. So Staphylococcus Of course, it's down here. I should have just like the capital first word and then lowercase second word, right? It's that simple. And when you're typing it, italicize it, please. Okay. So this is expected that you guys understand this, that it will be 2 words. The first name is the genus name. The second name is the specific epithet. That name together, Staphylococcus aureus, that's the species. Okay. That's the species name. But that second word is called the specific epithet. Together, written out, first one always capitalized. It's not just because it's the beginning of a sentence or something. Always capitalized, formal. And then the second word is not. And then you italicize everything if you're typing it. Okay. This can cost you points on stuff later on. So do remember that. You're going to see it so often that you're probably, it's going to become second nature to you. So FYI. Yeah, it says when you're in print, which is handwriting, you would underline it. I guess I'm not going to count you off for that one. Handwritten, I'm not going to count you off for that one. Typing is where it's going to get you. Okay. Taxonomy, classifying things, biological species. So it used to be just based on kind of what they look like and what traits they might have, right? So we've upped that now. We've upped our game now to including genomics, but so genes like your actual genetic makeup. With taxonomy, we can split it up into these major categories. You do need to know these, and you do need to know them in order. This is the most general, the domain, and this is the most specific, the species, okay? Domain, kingdom, phylum, class, order, family, genus, species. Whatever you need to do, make a little, anagram, whatever you want, whatever helps you memorize that, know them in order. Phylogeny, this is the taxonomic scheme, represents relatedness. This is based on evolution. So this is not just using genetics, but also the concept of maybe where we know that these guys fell in line as far as this guy's have existed for longer than this guy, so this is evolutionary, where we put it on the tree, and then we can determine that these guys here must have come after that because of these genetic changes. I don't know. I used to do some work in phylogeny, not particularly exciting. And then we have the 5 kingdoms, plants. There's no pathogens in there, by the way. Animals, protista. Protista includes algae. That's where algae falls with the protozoa. They sit right along with each other, even though they are not alike. And then we have Monera, and then the fungi. All right. Molecular genetics. This is just using genetics, DNA, so the actual genome of organisms to determine how related they are to one another. They use the small subunit of the ribosome, which I'll tell you what that means in a later chapter, but just real quick rundown, they're little parts in the cell that are going to help make protein. So if we go have your genes code for, something, your DNA codes for a specific protein. And then eventually we make the protein. The guy that's actually producing it is going to be the ribosome. So we can take the small subunit of that and compare it to the small subunit in other animals, because they all have to make proteins using ribosomes. It's how every cell works ever. And then we can see how related they are using that. So we've divided it up into bacteria, archaea, and eukarya using that information. Archaea, they look like a bacteria. They are not related to a bacteria. And so I guess my question for you guys, our question of the chapter, I would ask you guys as you're watching this video, what is a pathogen? And is every disease caused by a pathogen? So I don't know, think about that one. I mean, this isn't like some sort of, main thing, but just to get you guys thinking back on what we just learned. If you guys have any questions, email me. That's the best way to get in contact with me, or message me through Canvas. Both of those are great because I get an email and I can see that you tried to contact me. I'm not good about answering my phone. So FYI. And I'll see you guys for the next video. Awesome.